All right, good afternoon. Thank you for joining us for the Empirical Advances in the Neuroscience of Creativity session, the first part of the session. For those who were not in the room a few minutes ago, the first speaker in this session unfortunately could not make it today. So we will move on with the second session, the second presentation. With Ali Kelly from our lab. So Ali, take it away.
Connection problem for the people on Zoom. Just 1 second. We open again. We open it again, okay? I'll start that. Screen capture, close it. One minute, even less. Okay. Okay. This is this one?
Yes.
Okay. Almost done. Okay. Okay. It's okay.
All right. Thank you for your patience. So I'm going to be discussing neural differences in design fixation. As we know, design fixation is a type of functional fixedness. It's this phenomenon where if you are given a picture example and you're designing, you may reproduce elements of that design that you see in the example, even if you're explicitly told that some of those elements are problematic. And this phenomenon has been observed to be discipline specific. For example, mechanical engineers are more susceptible to this effect, as well as having some individual difference effects. And it's been extensively replicated, including in some more complex paradigms. And the question that we're asking in this project that I'll be talking about is, what is it about the pictorial example that biases problem solving in this way? And what are some of the neural mechanisms behind this phenomenon? And we're interested partly because in many fields, you have to teach using these pictorial examples, depending on your discipline. So the aim of this project is to establish a more clear framework for both the cognitive and neural processes that are implicated in design fixation, with an eye to potentially developing educational strategies that can shield designers from this fixation effect. The design, sorry about the text on this slide, but this is just to show some of the design of this project. It's quite comprehensive. We have a very rich data set for each participant, including extensive behavioral assessments, as well as brain imaging. But just wanted to note that today I will just be discussing one part of our brain imaging session, only the data that we have from our design tasks, both a control task and a fixation inducing task. So here are the tasks that participants receive. The control task is the coffee cup problem, where they're asked to design a coffee cup. You can see here that they're not given a picture example and not given many details about existing coffee cup designs. They're simply told to address specific problems that can occur with coffee cups, like leaking. In comparison, you see on the bottom the fixation condition, and here participants are asked to design a bike rack, but they're given a pictorial example of a present-day bike rack, including details about how it's mounted to the car. Also note that they're told some problematic elements of this design. For example, they're told that it's very difficult to mount bikes on this rack, and that the top mount design is an issue. In terms of the brain imaging setup, we have participants performing both of these design tasks in the scanner. They're using an MR-compatible tablet, so they're sketching their designs, and we're also having them think aloud while they're designing, so we have a lot of verbal protocol data for each participant. I'm able to record the session outside the scanner and monitor continuously. And we've had a lot of success training participants so that we have very little head motion and we get very clear audio. I'll try to play a short video here for you and we'll see if the audio works. We'll move forward, but we do get very nice data. And what we're doing with the data from both of these design problems is coding for both fixation. So on the left here, I know it's not legible, but this is a rubric that's been developed that we use to quantify the fixedness of each design that a participant produces. So for example, if they reuse an element from the bike rack that they're shown, for example, attaching it to the roof using suction cups, they'll receive a point. So more points in this schema means that they were more fixated. And here you can see just some examples of fixated versus non-fixated responses. On the bottom, you can see that the fixated response, they've produced a top mount design, and they've used similar mechanisms like a tire railing to hold the bicycle onto the top of the car. Just looking at behavioral data, we can see on the left, average fixation score by problem. We see this expected fixation effect. So in the fixation condition, the bike rack problem, participants are scoring much higher for fixation as compared to the control problem. On the right, you also see that participants are producing more designs. So fluency is higher in the control problem than in the fixation or experimental problem. The other thing we're doing with the data from both of these problems is categorizing it according to FBS, function behavior structure ontology, which is an ontology specific to the design process. This is a complex ontology, but I will just be focusing today on two aspects of this: R for requirement and S for structure. And what we do is we take each TR from our imaging session, and 3 experienced coders indicate which aspect of the FBS ontology the participant is focused on during that TR. So in terms of behavioral data, we can see that looking at structure, for example, participants in the fixation problem have more TRs that are classified as related to structure than participants when they're performing the control problem. Now, we'll just look briefly at some of the neuroimaging results from this project. So going back to that R or requirement portion of FBS, we see that when participants are examining or revisiting task requirements, so going back or speaking about, thinking about the requirements of the design problem, in the contrast for control relative to fixation, we see greater activity in superior frontal gyrus, middle frontal gyrus, anterior cingulate, so some executive regions, which makes sense with the idea that if you're thinking about goal-directed nature, how am I trying to fulfill the requirements of this task? However, it's interesting to note that we're seeing this more in the control, the coffee cup problem, than the fixation-inducing problem. And another interesting result is looking at that structure classification. We see increased activity again in the control relative to the fixation condition in inferior parietal lobule. So don't want to speak too specifically because data collection is still ongoing for this project, but one potential explanation for this effect where we're seeing this greater activity in the control relative to the fixation problem is that thinking about structure, for example, if a participant is simply copying an element of the structure from the example, so if they're just saying, okay, I'm going to attach this to the roof using suction cups, they're not engaging, they're not internally generating that element of the design, and so perhaps there's less neural mechanisms being recruited. So again, that was just a small taste of the analyses that we are planning to perform. There are a lot of other questions we hope to answer with this data, including can differences in participants' learning preferences help contribute to our understanding of what's going on neurally and cognitively with this fixation phenomenon? And why do we observe these individual as well as discipline-specific differences in fixation? So to be continued, and we'll have updates hopefully coming. And I'd just like to acknowledge the 2 principal investigators on this project, as well as our NSF funding.
Thanks so much, Ali. We don't have time for questions, but you can find Ali or any of us later on. So our next speaker is also our very own Christine Chesebrough. Christine.
Hi, everyone. I'm really excited to talk to you today about this big intracranial project I've been working on. I'm about a year in. Hopefully, there's a lot more data in the future, but we're really excited about what we've seen so far. So wanted to share it. So the project generally is about mind wandering and internal attention and trying to find the intracranial correlates of these sorts of internal task unrelated mind wandering category of cognition. So we know that mind wandering is important for creativity, right? We talk about that, but we know that mind wandering is not one thing. The nature of the cognitive state, as well as the sort of perceptual and epistemic demands of the stimulus that you're mind wandering away from, probably matter. In what context are you mind wandering and how might that help? Additionally, in mind wandering research, something that makes it challenging is that you need to interrupt people to ask them if they're mind wandering, and that just by nature interrupts the thought process. And so people are really interested in detecting attention states covertly, so where we wouldn't have to interrupt people. And I just wanted to point out this really great paper from Rebecca Rodriguez-Boerwinkle, who kind of really tore into the heart of the matter of the different complexities of the relationship between mind wandering and creativity. That was published in CRJ last year. So the ultimate goal of this project is to examine the intracranial correlates of transitions between internal and externally oriented attention during semi-naturalistic behavior. And I say semi-naturalistic because they're in a hospital bed, but they're watching movie clips. So that's naturalistic, right? So the patients are individuals with treatment-resistant epilepsy undergoing invasive inpatient monitoring. And while we start to collect data from patients with thought probes, we have like 5 years of data of people watching movies. And if you know anything about intracranial EEG, you know that it takes a long time to collect data. So we have this huge, rich data set. We have eye tracking. How far can we get? How far can we get if we just try to figure out when they're not paying attention to the movie from their eye features alone? So that's my postdoc. So how to find internal attention if you don't have any ground truth. So we know, what do we know about mind wandering? Both the spectral and oculometric signatures of internally oriented attention are task dependent. So they're different depending on the task. So we should only be trying to look to literature about videos or other visuospatial content. We also know that mind wandering is reflected in individually idiosyncratic deviations from expected behavior. So if your behavior does not reflect what would be expected from the task that you're supposed to be doing, you might not be paying attention. And group averages of responses to tasks likely, at least to some degree, approximate what the task is sort of eliciting. So if you're not doing what everyone else is doing, maybe you're not paying attention. But we also know that even if we know these things, there's lots of different kinds of mind wandering. The same eye states can be very different cognitive states, but we know what we might expect in the brain. We might expect sensory gating reflected in greater cortical alpha power, especially across lower frequency bands. We might expect some high frequency activity in the default mode network, and we might expect some DMN, frontoparietal control network coupling. So let's see. There is a rich literature on eye movements and internal and external attention. So we kind of know what we might be looking for. Fewer saccades, greater disparity of gaze, more pupil size variation, more blinks. We might see, are they focusing on the screen and are they looking where everyone else is looking? So there's lots of features to choose from. The data I'm looking at is from 11 patients out of 27 who had really good eye tracking data, where we didn't have to interpolate it at all. And they passively watched movies, and we had pretty good coverage across most brain networks, except really visual network, which is a limitation, but I'm comparing two different movies. Despicable Me, you may have heard of it. It's pretty entertaining, but people do mind wander during it. And then this other movie called Inscapes, which is kind of like a screensaver, and it's vibey, and it kind of feels like it's designed for mind wandering. So we did this in a hypothesis-driven way first. We've taken really careful steps because we don't know what we're looking at. But the net is that the hypothesis-driven first attempt actually matches what we see here pretty well with a data-driven approach. So let's just throw all of the eye features into a PCA and see what happens. We know what we would expect from the literature in terms of the loading based on what we expect more or less of. And wouldn't you know, but PCA found the same principal component in both movies, that if you look at the loadings and the direction of them, looks exactly like what we would have expected. Like I fell out of my chair. And then if you combine those feature spaces, you get that is the first PC. It looks like it might index internal attention. Saccade rate loads negatively onto it. Gaze disparity loads positively onto it. Gaze intersubject correlation loads negatively onto it. So is that internal attention? So it really looks like what we would have expected. So wow, things usually don't work out that nicely. But we also want to control for stimulus-driven responses. So are people doing what other people are doing? So we can just derive this basically Euclidean distance measure of, is the sum total of all your eye movements the same or different from what everyone else is doing? And if you're really far out, you're probably not mind wandering. If your eyes are doing basically what everyone else is doing, even if you're sort of doing internal attention, it might be stimulus driven. So I'll try to speed up here, but we used both of those things. So if both of those criteria were met, high loadings of that PC, and you weren't doing what everyone else was doing, we called that internal attention, and then we called the opposite of that external attention. And from that, we sort of picked out time points in each person's movie watching where we thought maybe they're internally or externally oriented. This paper just came out a few months ago, but it provides a really good model of sort of these shifts in low and high frequency activity between the DMN and dorsal attention network. In the default mode network, you expect sort of this internal to external shift to look like, I'm just going to show you the results. So in Despicable Me, we see some evidence of what we would expect. We see maybe greater default mode activity. We see greater cortical alpha generally in low frequencies. And we see that it is mostly corresponds with less high frequency activity, which these are usually anti-correlated with each other. But importantly, we see this default mode A and a negative higher default mode, lower dorsal attention. The better one is better, crazy, but we see really almost exactly what we would expect. Greater power in the default mode network A, less in dorsal attention, and we see these sort of patterns of greater connectivity between the frontoparietal control network and the default mode network A that we would have expected. So this was basically all very, careful, and we think that this is very promising. So there's similarities between these movies, but differences in key ways that matter for creativity. The internal attention in the Inscapes looks much more active. There's much more DMN activity. That's what we would expect. And so when we are mind wandering away from something that's sort of not very demanding, we might be engaging in a better kind of mind wandering. And that has implications for sort of when and how we might design mind wandering that's productive for creativity. And I will stop. Thank you so much.
Thank you so much, Christine. Again, you can find Christine for more questions. And our next speaker is Simone Lucien.
Okay. I want to briefly mention this work is part of an ongoing collaboration between my lab with Roger Beatty at Penn State and Emmanuel Voll's lab here at ICM. We're interested in studying semantic memory search and its relation to creativity. So looking at creativity, we know that it's supported by associative thinking, and specifically remote associative thinking, which involves linking distantly related concepts in memory. And the way that we would define how concepts are related and whether they are distantly related to each other is through semantic distance, which is a computational method through which you can tell the distance in meaning, the difference in meaning between pairs of words or sentences. But beyond remote associative thinking, we can look at the way that individuals search their memory. And if we understand semantic memory to be organized in clusters of semantically related concepts, we can understand that people can search their memory in one or two ways. One is clustering, wherein they retrieve successive concepts from the same semantic category. And the other one would be switching, wherein they retrieve concepts from different semantic categories during successive responses. And there's past work from Marcela, who is here at the conference, which has looked at the link between clustering and switching and creativity. And to highlight very briefly some findings, clustering seems to correlate with divergent thinking ability and to reflect a more persistent and goal-directed memory search, while switching correlates instead with convergent thinking ability, possibly reflects a more self-paced, flexible memory search. And importantly, both of these memory search modalities have a distinct relationship with brain connectivity. And we want to kind of follow up in this line of research. And the way that we do that is we collected 19 individuals who completed a word association task in fMRI, and we applied a pattern similarity analysis to the task activation. Now, pattern similarity analysis has the advantage of allowing you to study the patterns in brain activity, which goes beyond just magnitudes of activity or magnitudes of connectivity between brain regions. So it tells you essentially how the brain represents two different task conditions and how differently it might represent them. So, for example, when looking at an umbrella, the brain or an ROI within the brain will have a certain activity, a certain geometry to the brain activity, which will be different to when the individual might be viewing, say, a hand. And you can compare these two geometries of brain activation, and you can then determine how differently the brain represents these two task conditions, how differently viewing a hand is represented in the brain from viewing an umbrella. So this is a task that we assign participants, the associate diversity task. It's a polysemous word association task where participants are asked to generate creative word associations. Polysemous words are words that have a different meaning depending on the context that they appear in. So an example in English would be bank, because the bank, it could be the bank where you store your money in, or it could be the river bank, a geographical location of a river. And individuals were asked to first generate a creative word association to, say, the word bank. And then they were asked to generate a second word association again to bank by either clustering or switching, by staying in the same semantic category as the first association or to switch. They completed 30 trials by switching on their second association and 30 trials where they would cluster on their second association. We defined a number of anatomically individualized ROIs, spanning lateral occipital gyrus, pericalcarine gyrus, posterior cingulate cortex, the IFG pars triangularis portion, and the middle temporal gyrus, which spanned some brain networks which have been found to be relevant for creativity as well as clustering and switching in past research. And then we computed our representational distance scores within trial, importantly. So a single representational distance score essentially indicates how differently the brain represents generating the first creative word association, which is independent of clustering and switching because it is the first association. How differently the brain represents that to the second association when one is clustering or switching. So we generated two different distance scores, one for clustering trials where participants cluster and one for switching trials. And we did this for the ROIs separately and each participant. So here's the first analysis that we ran on the representational distance scores. We found that only in the bilateral inferior frontal gyrus, the pars triangularis portion, was the distance, this representational distance, higher in the switching trials as opposed to clustering trials. And what this indicated is that the bilateral IFG is sensitive to clustering and switching in some capacity, as it represents these more distantly, at least switching, it represents them more distantly to the first creative word associations as opposed to the clustering trials. We repeated the analysis, and this time we included semantic distance, which is a measure of remote associative thinking again. Semantic distance was computed between the association and the polysemous cue. And what we found was that the effect remains significant, but only for the right inferior frontal gyrus, which what this demonstrated is that the right inferior frontal gyrus is sensitive to clustering and switching beyond just the cognitive processes involved in remote associative thinking. Left IFG, in contrast, the effect became non-significant, which indicates that the left IFG is more so involved in remote associative thinking differences between these two trial conditions. So concluding, bilateral IFG is relevant for clustering and switching and memory search in general. And particularly the left IFG seems to be more specifically involved in remote associative thinking and how those cognitive processes might support clustering and switching. The right IFG, in contrast, certainly involved in remote associative thinking, but is involved in also cognitive processes beyond that. And this is what I have. Thank you very much.
Thank you. And our last speaker in this session is Coralie Ramos. Oops. Christine, do you have? Apologies for the mix-up.
So we're very much looking forward to that talk. Oh, it's the beginning one? These ones? Yes. Oh, okay, good. Is this
very good animation, but we can't see anything. So why can't we see anything? We'll try some magic.
And if you present it?
Yes, if we try to.
Oh, I
Nothing.
did just ask something.
But why?
Why? So that people wouldn't see me going and looking at the side.
Ah.
Yeah.
So now, okay, it's right. It's okay. So now, can we send it for the second? Okay.
Je pense que c'est parce que c'est une question de magie dans les doigts, il peut pas réagir.
Oh non, il est chiant. Hein. C'est même instable et tout. Il y a tellement de gens qui le tripotent que.
Oui, c'est vrai qu'il est pas mal. Ok. Merci. Can we test?
All right, for real this time.
Okay.
Coralie Ramos, a warm welcome.
So I'm very delighted to be here with you today to present this wonderful study exploring the dynamic rewiring of electrophysiological brain networks during creative thinking. Brain imaging, like fMRI, have helped us understand that the brain is a network of connected areas that work together and communicate over time to support thinking. Here, EEGs can help with their higher temporal resolution to track quick switches in brain activity. The first step of this study was to investigate if creativity can be characterized as an individualizable cognitive function. So we asked healthy subjects to do 2 tasks, one creative and the control. From their data, we identified different brain network states. This allowed us for the first time to compare how the brain states shift over time between creative and non-creative tasks, permitting us to explore whether creativity brings the brain into the same states differently organized in time, or if creativity brings the brain into unique brain network states. The second step was to look at creativity scores to see how dynamic brain network states can influence individual creative cognition. So by focusing on how brain activity changes dynamically during creativity, this study takes an important step toward understanding the brain mechanism behind creative thoughts and its stages. 93 subjects completed the 2 tasks while their brain activity was recorded in high-density EEG. During AUT, subjects had to imagine as many unusual uses as possible for 20 objects, while in OCT, they had to focus on their typical features and physical properties. So both tasks required attention and memory, but only the creative task added some processes to achieve something new and original. Then some pre-processing shifts were assessed, and we used a method called EEG source connectivity combined with a sliding window approach to study the brain's functional connectivity. For the brain state analysis, all matrices were concatenated to extract by a clustering approach strong global patterns appearing over time, the BNS. Then we backfitted them on individual matrices, allowing us to reconstruct how each participant's brain moved between states during the task. From this, we calculated some temporal metrics to describe how these brain networks behaved. So our results identified 5 distinct brain network states, labeled BNS 1 to 5. And we found that their distribution was clearly different between the creative and control conditions. Specifically, BNS 1 and 2 appeared almost exclusively during the control task, while BNS 3, 4, and 5 appeared mainly during creativity. Creative and non-creative task states show distinct patterns of activation and connectivity. Creative states are characterized by a more distributed involvement of brain regions, while non-creative states are essentially with frontal activation patterns. One important finding is that the Richmond cingulate cortex, a keynote of the DMN, was only active during the creative thinking and was one of the most active regions overall. Looking closer at the DMN, creative states involve more nodes, which established more connections. We also noticed a clear difference in how stable or variable brain activity was. Control states are homogeneous, showing always the same pattern of highly active regions, while creative states are heterogeneous, suggesting more flexible and dynamic thinking. We also looked at how different brain networks communicate with each other. In creative states, we saw stronger and broader communication between the DMN, the FTN, and the DIN. Importantly, we only found in BNS 3 and 4 a reciprocal communication between these networks. And interestingly, during the control task, we saw more connections between the DIN and the DMN. To go further, we divided participants into high and low originality groups and compared their brain network dynamics. For BNS 5, we observed the negative trends. People who spend more time in these states tend to have lower originality scores. This trend was confirmed by group comparisons, showing significantly higher BNS 5 values in the low originality group. In contrast, for the BNS 4, we found the positive trends. People in the high originality group spent more time in these states, and this engagement is correlated with higher creativity. To better understand how these creative states evolve over time, we looked at the transitions between them. We found that, as a stereotypes way, the low originality group exhibits significantly higher transition from BNS5 to BNS3, and the probability of this transition is also correlated with originality scores. Finally, we looked at entropy, which tells us how diverse or flexible the transitions were, and the high originality group had higher transition entropy from BNS5, meaning they showed more varied and flexible switching to other creative states. So to discuss, we hypothesized creative states reflect different steps of the creative process. State 5 represents the idea exploration. It involves deep internal generation with strong activation of in posterior brain regions, like the isthmus cingulate cortex, but spending too much time in this state does not allow an efficiently creativity. State 3 represents the idea integration. It involves different brain networks like the DMN, the FTN, and the DIN, and all communicate together. And state 4 represents the idea evaluation. It involves mostly frontal activity, especially executive control regions, and spending time in these states is necessary to allow originality success. Also, the study highlights the importance of the neural flexibility, the ability to move dynamically between exploration, integration, and evaluation. And altogether, our results support a dynamic network-based model of creativity, where creative thinking starts with spontaneous idea generation, moves into integration, and is refined through evaluation. Thank you for your attention.
Thank you so much, Coralie, and to all the speakers. So we'll take a very, very brief, less than 5 minutes break as the speakers for the second part of the session transition to the front. So stretch your legs, get a cup of coffee, but come back here sooner than later. So thank you, everyone. Another round of applause for the speakers.
All right, so I'm gonna say welcome to the next. Oh,
Yeah.
Okay. Hi guys, we're a little behind schedule, and I'm sure everybody is ready to go find some coffee. So I thought we should get started, and this is the second panel on our second session on empirical advances, and our first speaker in the session is Carola Salvi.
Thank you. So it looks like I'm going to show you some of the colorful pictures that Wendy just mentioned about. Okay, so here's a brief paragraph that captures some of the key aspects of insights that were unveiled thanks to neuroscientific studies. Okay, we're not going to go through them all, but today I'm going to show you some new results using diffusion tension imaging. So one of the things that I want to point out right away is that when we study the neural correlates of insights, we use this kind of puzzles or like remote associates, anagrams, so on and so forth, but foremost for methodological reasons. Why? Because they're short, they're compact, so we can give a lot of this in a session while people are, for example, in a scanner. And that does increase our statistical power. So the scientific journey of insight problem solving began with a broad question about hemispheric difference. And in their seminal work, Boden and Beeman found that there was faster recognition for problem solution when they were presented to the left visual hemifield, implying that the right hemisphere has a functional advantage in problem solving. Follow-up studies using EEG found that there was gamma activation over the right anterior temporal lobe right before people press the button and declare that just have an insight. The way we measure insight is by giving those kind of riddles, and people press a button and then they have to decide if they solve a problem with insight or via step-by-step analysis. This brain region was better localized in an imaging study over the right superior temporal gyrus, and a more causal approach using TDCS found that if we stimulate this brain area, people tend to solve more problems via insight. So the right superior temporal gyrus seems to be a hub that facilitates processing of distance associate relationship. This makes sense if we think that insight implies a combination or recombination of existing representation in order to generate novel ideas. These novel associations tend to be more original and therefore creative. If we look in the literature of hemispheric differences, we do find, in creativity in general, we do find that lesions to the right hemisphere are associated with impaired creativity, whereas damage to the left hemisphere tend to be associated with like somewhat increased creativity, implying what people call a releasing effect on creativity. Again, if we look at other measures like artistic creativity, it seems to be associated with decreased thickness over the left superior temporal gyrus, which is known also as the Wernicke area. To summarize the big literature on hemispheric differences, we can kind of say that prior findings implying that the left hemisphere regions do play a regulatory role in creativity, and their disruption seems to promote novel ideas. This is why we decided to look at DTI. What is DTI? What is this technique? So basically the idea is that we are looking at how healthy are the tracks in the brain that connect different brain areas. So this is like made of like white matter, okay? And what we measure is the diffusion of water molecules. To make it super simple, when I'm gonna show you the results, basically we are looking at two different variables that are inversely related. One is fraction anisotropy, and this reflects better white matter integrity, meaning that a high fractional anisotropy is good, means that the fibers are healthy, and it's inversely related to mean diffusivity, which measure the average rate of water diffusion in the tissue. If we look at the literature on divergent thinking and DTI, since like there aren't really studies on insight, what we find are like two different lines of research. And this is kind of like what I read and what like helped us framing our hypothesis. So it seems that there is a whole line of research that found negative correlation between divergent thinking and fractional anisotropy, foremost to the left hemisphere. Whereas by contrast, there is another line of research that instead find positive correlation between fractional anisotropy and divergent thinking. And these results seems to be led by gender differences. So I pointed that out there. So that's why in our study, all of the results that I'm going to show you are basically controlled for sex and, of course, age. We administer 6 compound remote associates to our sample size. And here's the results. So we do find that insight was predicted by lower fractional anisotropy, like the first line of research that I show you in the former slide, over the left posterior arcuate fasciculus. You can see it there. And another thing that I want to point out is how these measures, reflect, the inverse relationship that there is between FA and MD. This is quite relevant for the quality of the results. So this brain tracts does connect the left superior temporal gyrus with the angular gyrus. So it's the other side of the brain compared to what we found being more active when people have an insight. Where we also found that insight was predicted by lower FA, one more time, in both left and right superior longitudinal fasciculus. This is a brain tracts that connect the parietal cortex to the frontal lobes, okay? Specifically the inferior frontal gyrus that Simone already explained you what it is, so I'll move forward to save some time. So what can we conclude from these early results, okay? Is that it seems that lower fractional anisotropy in these regions of the left hemisphere seems to have a role in generating ideas via insights. How did we interpret it? That this might diminish competition between the two hemispheres, okay? Inhibiting strongly activated information over the left hemisphere, probably to favor associative thinking that it's happening, okay, over the right hemisphere. We also found results that are associated with non-insight solutions when people have this kind of like analytic kind of thinking. Specifically, what we found is that there was higher fraction anisotropy over the left VOF, vertical occipital fasciculus. This is like a fasciculus over the visual cortex, okay? And we also found that higher fraction anisotropy predicted step-by-step analytic problem solving over the anterior corpus callosum, which is, you know, this bundle of tracts that are in our frontal lobes, okay? And the idea is that, one more time, this result does reflect enhanced executive functioning. So while we were like interpreting these results, we did find that there was one study on convergent thinking that found the same results that we are looking at, that matches us. So we started like thinking, and then I'm going to probably like leave you with a question instead of with just some results. That basically what we were doing, we were comparing a lot of results on divergent thinking using divergent thinking task that somehow did match or did not match our results. But one of the points that I would like to make is that if that was a fair comparison that we were doing, okay, comparing divergent thinking task with what we were getting when we were measuring insights, it could be, but I'm not sure, that people, and that's why we find some overlaps or not, but when they were doing the divergent thinking tasks, sometimes they were solving those problems via insights and sometimes they didn't. So I'm leaving you with the question if we can actually measure insight in divergent thinking task, and this could be an interpretation of our results.
Thanks, Carola. Our next speaker is Karina Perktold Stephan. Hope I did okay with that name.
It's fine, thank you. All right, welcome to my talk. I would like to discuss today if men use fists and women use whispers, which is a very ominous reference to gender differences in malevolent creativity. So first, what is malevolent creativity? It is the generation or implementation of creative ideas in order to intentionally hurt others. Think about creative lying, manipulation, or even unique crimes. And here we could look at individual differences. So who's malevolently creative? Or we could look at situational factors that increase malevolent creativity, ideally both. In terms of assessment, we could ask people to self-report their malevolent creativity behavior, or we could put their abilities to the test and have them generate many creative ideas how to get revenge on others, which is what we like to do at the University of Graz. And today, I present you an individual differences perspective because I'm asking the question if men and women differ in their capacity for creative harm. Now, you may think, is this some low-hanging fruit? And I'm picking it because, yeah, it is. But actually, in the past, a lot of publications on malevolent creativity have consistently addressed gender differences, asked how important they are, and we've used to discuss them as limitations. So I want to know, is that really that big of an issue that we always need to address? Yes. So here's an item of our malevolent creativity test with a repertoire of many creative ideas that real participants have generated, and I've had contact with these participants, so I'm a very brave soul. In this situation, you have to take revenge on a neighbor that has been unfair to you, and you're trying to get back at them for not paying you for your work. If you have a look at these ideas, I think we have a very broad repertoire of creative craziness here. So what we're trying to do is right now we're trying to reanalyze our past research data from the past years to see if there's a merit to gender differences in these ideas. The whole plan is to analyze 5 studies with around 750 participants, but today I'm presenting 4 with around 600 participants. And what we're doing is we're looking at gender differences in various indices of malevolent creativity potential on our test. So fluency, harmfulness or malevolence of ideas, originality of ideas and a total score of malevolent creativity. But we also dive deeper into quality. So we want to know, are there gender differences in the types of aggression that people use? So for example, in physical aggression, harming others, property aggression, destroying their property, social aggression, lying, manipulating, threat-related aggression, stalking, harassment, or tricks or pranks. And we're not only going to look at gender differences at the level of the behavior, but also on the level of the brain. So today I'm presenting some exploratory findings on resting state chain gender differences in brain connectivity. So now on to our preliminary results. In our first study, which is not at all looking nicely on this slide, so I hope it gets better. The only difference we found is for ideational fluency, in terms that women generated more ideas than men on our task, but overall no other differences emerging. In study 2, the fluency effect did not replicate, so that's good, right? But men generated ideas of higher malevolence, so more harmful ideas than women. And there was a tendency for men using more physical aggression in their ideas compared to women. Now, in our third study, the malevolence effect replicated. So again, men showed higher harmfulness of ideas compared to women, and at a trend, higher originality this time in our study. And there was again a trend for men using more physical aggression, and this time women tendentially implementing more social aggression in their malevolent creativity. In study 4, again, the pattern was rather diverse. We managed to replicate the previous fluency effect in favor of women. The malevolence effect for men was only significant at a trend, as was the effect for more physical aggression in men. So overall, I would say it's a mixed bag of findings. Effects are weak. So what do we do if behavior is inconsistent? We move to the brain, of course. So at the first step, we ran a multivariate pattern analysis to see if malevolent creativity is generally related to resting state functional connectivity of the brain. We did not find significant patterns in general for malevolence and originality, but we found connectivity patterns for ideational fluency and for total malevolent creativity. Ideational fluency was linked to connectivity, for example, of the occipital pole, left frontal cortex, but also the lingual gyrus. And the composite score of malevolent creativity was linked to connectivity of the left temporal pole with the rest of the brain. This is interesting, but it gets even more interesting when we look at gender differences, because for malevolence, the pattern for men and women was rather different. So for men, malevolence was linked to connectivity of the precentral and postcentral gyrus, but also the left inferior frontal gyrus, while for women, it was only linked to connectivity of the right parahippocampal gyrus. For originality, differences again emerged. For men, it was linked to postcentral, precentral gyrus connectivity, also the left temporal fusiform gyrus and parahippocampal gyrus. For women, seems to be again a difference, right parahippocampal and left supramarginal gyrus with the rest of the brain. Now, these are rough preliminary analysis. Right now, we're running post-hoc analysis to see in which direction the connectivity actually goes, and if the results remain robust with more strict corrections, of course. But it seems to be the case, although they're not really prominent behavioral differences, there are some interesting differences in how men and women go about malevolent creativity. And this is very exploratory, but as Carola already explained before, there's also like diffusion tensor imaging, which we're also using in our study. So we want to know, is malevolent creativity linked to the integrity of the white matter fiber tracts? And there's some very beautiful brain pictures that I'm still working on interpreting. We have links of malevolent creativity to the associate cingulum linking frontal parietal areas, left and right cerebellum, but also like arcuate, left arcuate fasciculus. And we have preliminarily gender differences. So for women, quantitative anisotropy of the left parafactory cingulum seems to be more important, and also like links between frontoparietal areas. For men, interestingly, it's more like the interhemispheric connectivity of the corpus callosum and the right arcuate fasciculus. So I'm still working on interpretations. I think it's promising, but overall, to quickly sum it up, so I think evidence for gender differences in terms of behavioral output of malevolent creativity are rather weak and inconsistent. We replicated some effects, but they're not really convincing enough, I think, to make this a bigger issue than it is. But it is interesting to look at the level of the brain. And this nicely ties into previous research on gender differences in creativity overall, right? So the general gist is men and women don't differ in their creative output, but they use different strategies and neuronal mechanisms to be creative, which is a nice idea, I think. So if you're interested in the dark stuff, do get in touch with me, and thank you for your attention.
Our next speaker is Yuhua Yu.
All right. I'm going to deliver my takeaway first in case you're tired or I cannot finish in time. Just like the stock market, high volatility hurts performance. The same thing goes with neurovolatility in the brain when creativity is concerned. So first, I would like to thank my mentor and collaborators who contributed to the 2 studies I'm going to present today. Neural oscillations observed in EEG data has been implicated in all aspects of cognition. The power of those oscillations, indicating the degree of oscillatory synchronization, modulate all sorts of things, including attention, executive function, memory processes. So if we think of these power are like knobs that people can use to control cognition, then how fast one can turn up and down these knobs have to bear some consequence on the behavior. So if the power changes slowly, namely low volatility, that's indicating some sort of oscillatory stability. And if the power change fast, that means more high volatility, indicating more rapid transition between synchrony and desynchrony. So in the past publication, we did manage to associate these volatility with people's problem-solving performance. We had people solve those compound remote associates, as Carola has introduced. We used exactly the same problem to see our common origin there. We then group those trials based on the outcome. So does a person solve the problem or not? And if they solve the problem, how they solve the problem. And then we looked at the volatility during the solving period and compare that across the different types of trials. It turns out that if a brain had a lower volatility during the solving period, the participant is more likely to solve the problem with insight, also those aha moments, compared to solving with analysis. But if the volatility is a bit higher, they tend to report an incorrect solution. And if the volatility is even higher, they didn't even report a solution at all. The trial get timed out. And this is not only true for the solving period volatility, but also true for the pre-onset volatility. That is, the volatility during the 1 second window, even before the person sees the problem, can predict the outcome of the trial in exactly the same order. And we replicate all these findings in a different task, these anagram problems. So here I want to focus on this pre-onset effect. That's really fascinating. So this is saying the volatility during this your mental state you were in during the preparation can actually predict if you were in the lower volatility, you are more likely to solve the problem than not solving the problem. Now, even though this analysis was done all as within subject comparisons of statistics, but we start to wonder, does the individual difference in the volatility may also predict your problem-solving ability? So luckily, in this large project, we actually collected resting state EEG data from the same group of people, and they were collected on separate days from those anagram and the CRA problems. Each resting state has 10 minutes of EEG data with alternating between eyes open and eyes closed condition. Now, briefly, how we compute volatility, we first extract the power envelopes from the denoised EEG signals in these classically defined frequency bands. Then we apply the volatility formula to each power envelope, which is simply the standard deviation of the step-by-step change in the power. This gives us volatility for each frequency band at each electrodes. And then we just average across to get a single volatility value. Now, first, we notice that volatility turns out to be stable within a person. So if someone has high volatility during resting state, they also tend to have higher volatility during the problem solving. We also find that volatility are modulated by what the persons are doing. You tend to have higher volatility during rest compared to performing a task. But within the problem-solving session, you actually had lower volatility during the short preparation period compared to really actively solving the problem. Now, the more important question is, does the resting state volatility really predict the person's problem-solving ability? we had some evidence for that. It turns out that if you have lower volatility during the eyes closed rest session, the person tend to solve more problems, both in the anagram and in the CRAs. We did not have a significant effect for the eyes open volatility. So far we have some findings that volatility is indeed associated at both inter and intra-individual level with the problem-solving performance. But does all that generalize to idea generation? So for this question, we have a new study. We use those metaphor generation tasks, and when I was already mentioned also. Each trial, you first listen to a short description of a science concept, like allele is one possible version of a gene, and then you are asked to generate a metaphor for allele. What allele is like what, with EEG data recorded. Then we collect all those metaphor responses. We have a separate group of people, these are our crowd judges, to rate the creative quality of each metaphor response, how novel the metaphor is and how apt the metaphor is. So the main question is, does the creative quality of those metaphor responses are associated with the brain volatility during the generation? So first, let's look at the metaphor novelty. Now, because in each trial, the person has to go through different stages to generate the metaphor. They first have to listen and processing the concept, and then actively thinking about the metaphor. So we separate a trial period into two segments, an early encoding period and the late generation period. So we're going to look at the volatility in these two periods separately. It turns out that if the brain has lower volatility during the encoding period, the metaphor the person generates will be rated as a high novelty by other people. So again, a negative association. But we did not have a significant effect for the generation period volatility. Exactly same pattern for aptness, right? Again, lower volatility during the encoding period is associated with a higher apt, more apt metaphor response. So to sum up, we find that lower volatility indexing more stable neural oscillations are associated with better creative performance at both within and between subject level and in both problem solving and idea generation tasks. Thank you.
No, I think we have to, yeah, but find Yuhua later. The last speaker is Orhan de Manzano.
All right, last 7 minutes before your date with Dionysos. Take a deep breath, move around a little bit. We can do this. I will keep it light. I'm going to pick up where Fredrik Ullén left off this morning, talking about data we collected in collaboration with the Swedish Twin Registry. It's quite a large sample. Here I will be focusing on creative achievement and divergent thinking, and some control variables in this case that we collected earlier. I wanted to have a divergent thinking test implemented already in 2012, but I was denied, partly rightfully so, because I didn't have any good idea for exactly what task to use online unadministered. But since then, there's been a lot of progress in using semantic distance as a proxy for creative ability. And in 21, there was this test published, which is very simple. You just generate 10 nouns that are as different from each other as possible. You calculate semantic distance between the words, and then the authors found low to moderate correlations with, well, traditional creativity, well, psychometric creativity tests. And this is something that could be used with the twins, we thought. And this sort of generally supports the associative theories, you know, joining the choir of people saying that creative people are better at linking remote concepts. They didn't, however, study the relation between this task and real-life creative achievement, which would be relevant if we want to study the nature and nurture, I think, of this thing, even though we should expect such an association. So firstly, well, I will talk about two data sets. Firstly, then the web survey data, and then also an MR data set using a very similar task. So firstly, yes, we have the web survey data, we have the DAT. We looked at the maximum creative achievement across 7 domains. and a decent sample size. And skipping directly to the results, people who produce less semantically similar words tend to have higher real-life creative achievement. No surprise there. We find a similar relation for artistic and scientific creative achievement. This is probably the prettiest graph I will ever present. So, remind me to always have 8,000 participants. Jumping, oh yeah, and then we did a ordinal logistic regression here just to show that this measure is still sort of relevant even if you put in some other factors that are known to be associated with maximum creative achievement. So we have sort of established the first thing, which allows us then to jump to the MR data. So we had 63 twin participants who did a skill learning task. They were taught to play the piano over 6 weeks. And again, I managed this time to lobby for a divergent thinking task to be included. And we also had data from 17 non-twin participants. So for the abstract, I think I only included the twin participants. And during the last weekend, I, in a masochistic fit, also entered the 17 pilots. So we have now 80 participants in total. So I can show you sort of the latest news. The task is somewhat similar. I guess it has been compressed a little bit here. Basically, it involves generating close words and remote words. We also had a control task where they're supposed to remember close and remote words, but I will not go into so much to that. So jumping here to kind of the sanity check, they did produce more remote words during remote. And also interestingly, in this small data set, we did find that semantic similarity was negatively correlated with creative achievement. In this case, probably depending on the composition of the sample, only scientific creative achievement. And also I could mention that there was a relation with psychosis proneness and generating remote words around the same. It was like mine, yeah, 0.34. Okay, so in this, let's see, whoa, hang on one more time, again. Yeah, so in this lo-fi presentation of the results, you see a prefrontal cluster of the inferior middle frontal gyrus, you see the dorsal premotor regions, and you see a large occipital cluster with a peak in the lingual gyri, going forward into the temporal cortices through the fusiform and dorsally, medially, and to the superior. So that's looks like what you would expect and what others have sort of seen. We also did a parametric modulation here, looking at activity that co-varies with the remoteness of generated words, controlling for activity that remains constant throughout the trial. So that's going to be pretty specific. And here we see then really the left and right lingual gyri. So this is very interesting, and this has popped up previously in other studies, and I think it's about time we try to figure out what is actually going on in this region, since it keeps popping up both in generation and evaluation, as shown from here, of semantic similarity. And since there will be no questions, I presume, before the wine and cheese, I would invite you to come and see the poster of Jessica White, my student assistant, who is presenting, yeah, there she is waving, who is presenting a study on verbal divergent thinking and then eye behavior. And oh, how interesting, you say. And I agree. And so, yeah, others people have shown this. We're sort of replicating things. We're really zooming in on this region. And that gives, I think, a good reason also to extend on the research to ask this question then, do twin siblings show more similar brain activity patterns during creative performance? And the short answer is yes, they do. And we will continue to work on this. I don't have time to go into all the details. We'll continue with twin modeling and doing all sorts of interesting stuff that then we can continue to discuss at poster 32. Okay. Thank you.
All right, that wraps up the last talk session for today. We have, as was mentioned, we have wine and cheese. We have posters, not incidentally right after the wine and cheese. We invite you to dance. We have an interactive dance session that'll be right across the way here in room 1-2. See you there.